going on, trying to kind of focus more on the things I think of as more as like extension related, and then kind of upcoming projects, assuming things get funded, and areas I'd like to be able to work in the future, and some of the sort of additional activities that I do in the B Lab currently. So I feel kind of bad, especially making this slide. I think of all these people I've seen give talks like this, and they have some interesting background of where they came from some far off place, and how they managed to get here. But I was born in Washington, and so I've gone like a whole, I don't know, 100 miles or something from where I was like born and raised. So I was born in 9 Mile Falls, right along, I grew up on the Spokane River, just north of Spokane. And then I attended Eastern Washington University, where I got my undergraduate degree in education. So originally I was going to be a high school science teacher, and I actually worked part-time for an alternative high school in Spokane Valley while I got my master's degree at Eastern in science. And then I came here and got my PhD at WSU in Dr. Steve Shepard's lab, and did a postdoc there for a couple years before I moved into this current position that I'm in. So at Eastern Washington University, I mainly worked on reproductive techniques, mainly on cryopreservation. So the background on this slide here is honeybee sperm that's been stained, and this is a fluorescent microscope. So the green cells there are alive, or at least the membranes are intact, and the red cells are dead. And so I mainly worked on like frogs and mice for the first like almost a year and a half of my master's degree. And then really right at the end, we worked a little bit with Steve Shepard and Sue Covey on honeybee semen freezing. And they had just gotten a permit to import honeybee semen at that time. And so we're really interested in the ability to bring semen into the US, like frozen, and store it both for, posterity's sake, and for breeding. And so nobody was really doing this at the time. There'd been a few papers in the 80s, but nobody had successfully been able to freeze honeybee semen in a way that they put it into practice. And so that's what I kind of started as my PhD at WSU, was trying to develop a practical method that could be implemented. And so the idea is to be able to freeze semen. That's what in this tank here, be able to thaw that semen, inseminate a queen, produce daughters, and then thaw again, inseminate those daughters. And so you continue to do these back crosses from the tank, because it's been illegal to import honeybees into the US since 1922. We haven't been able to import any new or novel genetics since then. And with this permit was restricted to only honeybee semen, so we couldn't bring in eggs or anything from the maternal side. And so, but frozen semen still allows you to do enough back crosses that you can often get very close to the original line that we were trying to get. And so while I was didn't go far from where I was born here in Washington, the honeybee world and this project has allowed me to do quite a bit of traveling. So as soon as I started my PhD, kind of first thing I did was demonstrate and publish a paper that you could do these sequential generations by freezing semen, thawing it, inseminating daughters after daughter. And so I started going on these trips to collect honeybee semen and freeze it. And so starting in 2011 or 2010, we have gone every year until COVID, really. And so places like the Republic of Georgia, like these Caucasian honeybees from the Caucasus Mountains, multiple trips to Italy for Ligustica, the Italian honeybee, France, Slovenia, Kazakhstan, to collect honeybee semen for freezing. And so this picture is the tank here, where we have sort of the first germplasm repository for honeybees. And then in like 2014, we worked with the National Animal Germplasm Program in Fort Collins, Colorado, to include honeybees, the first contribution of honeybees to that National Animal Germplasm Repository. And so, I mean, to me, that was very cool, because instead of, I think a lot of the work, sometimes people maybe publish papers and then they don't actually do anything with it. It kind of doesn't ever get implemented. And so it was nice to work on something that maybe is not necessarily seen as very applied or practical, but then to take it all the way from the sort of development scientific side and then put it into practice and have it be something that's used by the industry and put into practice. It was pretty exciting. And I say that because some of you might know Eric Olson, um, uh, was a beekeeper, still an orchardist. When I first met him as a student, Steve introduced me. He asked what I was working on. I said, freezing honeybee semen. And he said, oh, that's great. Well, maybe once you are working in here in the lab for a little bit and get your feet wet, maybe you'll work on something important. And so that was the very first conversation I ever had with him. And so I took that to heart and remembered it. And, as I worked in this field longer, I understood that more and more. But I do think this does have, practical implications, and hopefully we'll have like an impact in the industry in the long term anyways. And I do think, working with him, and he still calls every week at 6 a.m. Did help me, and you'll see hopefully later, work on more immediate and practical projects as well, which I equally love. And so here's just a few of the members. Riley Reed, I guess it's still your left also, but with the bee beard on, and Sam Dilday are PhD students. Sam is just starting this fall, and Wesley Hester is the new apiary manager. And I also have another master's student coming this fall as well. I have 2 postdocs. I had to change this slide just, within the last couple of weeks. I did have 3 postdocs. So Kelly Kulhanek, who was hired as the new assistant professor in entomology on honeybees, was my 3rd postdoc. But Gus Zhang and Ray Olson, postdocs that work on a project I'm not going to talk a lot about here, but is a trade assistance for specialty crops. It's a big project following colonies through multiple pollination seasons and looking at bee health and pesticide residues, and a really big, pretty cool project. So put this over here, publication record, sort of just to give you, for those of you who don't want to dissect the CV sort of thing. I did get some advice from someone here in the department. So this is a cumulative, right, year by year. So this isn't like the number of pubs per year. This is a cumulative, and he said that way it looks much better and like you have more publications. But I have been working hard to increase my publication rate each year, and I've been pretty successful in increasing the number of pubs per year over the last 3 or 4 years. And similar with the grant funding. And the grants that I receive are a mix of these federal and state grants, like the specialty block grant, some WSDA money. But like I said, that trade assistance for specialty crops. I have a couple of these AFRI and NIFA grants, and I'll talk about some that are pending as well. But I have a larger number, but less money that come from industry groups like the California State Beekeepers Association, the Idaho Honey Industry, Project Apis M, and some of these other funding groups as well that are not federal or state grants. So my current sort of generalized research areas are in IPM as they apply really to Varroa management and generally commercial management practices. So, and not that I don't work with hobby beekeepers, but a majority of my work is really on large scale commercial management and with commercial beekeepers. And those kind of fall into these 2 areas of indoor storage, which I'll talk about today, and then sort of general pollinator health during
and also
pollination and how they move
some of them into other stuff.
What is that? Hey, Steve, will you mute your?
Oh, I'm so sorry. Yeah.
And so, I generally don't do a lot of insecticide work, but like I said, that the trade assistance for specialty crops project involves quite a bit of insecticide testing of the pollen that the bees collect. And then we have some other projects that involve the pollinator health during blueberry pollination. And Riley Reed, my PhD student, is working on some pollination issues in seed crops. And then nutrition is sort of a newer one and really just a sponsored research project, but has got me involved in supplemental nutrition for honeybees. Uh-oh. Some PowerPoint crashed on me. I haven't had that happen. Well, it says it's coming back on. I don't know.
So it crashed on your computer.
Yeah, it just quit and said unexpectedly quit. If it won't come back up, I can
Relaunch it.
from my laptop or something.
You want to try relaunching it though on the machine you're on?
Yeah, let's see here. And then I think I need to reshare first here.
There you go.
Okay. Does that work?
Yep.
So a majority, a big part of the program right now deals with the indoor storage of honeybee colonies in the winter months, but also some of the sort of new research that we're doing involves the use of these storage facilities and how to use them in this commercial management IPM strategy, and using the storage space to put bees in at times other than winter. And some of the outputs of this was a big part of it is this booklet. This is the cover of the booklet with funding from this Healthy Hives 2020 initiative and Project APSM. We published this booklet in 2020. This was the first edition. And then since then, I'll show you in the next couple of slides, we have a second edition that's online and it's much cooler and kind of more interactive. But for those of you that don't know about this practice, it's relatively new in the US. It's something that beekeepers in Canada have done for a long time, which is to try to protect their colonies from these cold, harsh winters. They put them in storage buildings. But it's really caught on here in the US, really for different purposes, not necessarily to protect them from the cold. In some ways, it's almost to protect them from warmer winters, and also as a strategy to kind of keep them out of California as long as possible before almond pollination. So this figure here just shows the number of colonies that move through California border inspection stations. So these, and I think this is in, yeah. So these are colonies that move through these inspection stations in January. And so it shows that Idaho has the largest number of colonies, more than 300,000 colonies going into California in January. And so probably the number of colonies that are native, say, to Idaho, that is probably 80,000 or something like that. So a huge number of these colonies. The reason why so many colonies are coming out of Idaho and North Dakota in January is because of this new practice of storing bees indoors. And so as part of this booklet, my postdoc Kelly Kulhanek and I surveyed beekeepers in these different states and generated this figure. And so this is sort of the first survey or accounting of the number of colonies that are stored indoors and where those colonies are. And so here you can see as of last year, almost 500,000 colonies are stored in buildings in Idaho. And it's amazing, like in Idaho, for example, one of the new buildings there in Idaho, a single building holds 82,000 colonies under refrigerated storage for 3 or 4 months before almond pollination. So it's a pretty cool strategy, and beekeepers are finding a lot of success with this management practice. And so this is the cover. I thought it'd be kind of too complicated. I was gonna click on it and kind of show you guys the online format for this, which is kind of neat and flashy. Instead, last minute, I just pulled some screenshots, but this is also available in a PDF format. But the main idea was to have it online so it could be updated continuously. As part of this, we also did a conference in 2021 and had a few different speakers and different beekeepers who practice this storage technique talk. And that conference is published online, the video of it, it's on YouTube. I think as of today, when I pulled the screenshot, it had a little over 1,000 views. And then as part of and in preparation for this conference, Kelly and I and the video folks from Project APSM, which is where the funding came from, went and shot a series of these 4 videos that covered different aspects of the indoor storage. So putting bees in there, you know, how the HVAC systems work, how to handle the bees before and after, that kind of thing. And so these, you know, combined have, you know, a little over 3,000 views of these 4 sort of short videos associated with indoor wintering. And so the sort of research component of that still continues. So we use these refrigerated cargo containers, which are over at the Othello, you know, potato farm, the irrigated research farm near Othello. And they're being moved over to the honeybee Othello lab here next week, actually. And those are going to be updated with some new refrigeration systems. Backwards. Okay, and oh man, PowerPoint is really struggling today. Okay, so that's some of that indoor storage, and there's a number of products. We just submitted a paper on storing banking queens indoors during the summer as a strategy to help mitigate these very hot summers in California, where a lot of the queen production occurs. And so they bank a lot of queens in July and August in Northern California, and the very high temperatures, increased incidence of wildfire and stuff like that makes it very difficult often to bank, you know, tens of thousands of queens for these big commercial queen producers in Northern California. And so we had some pretty good results with banking queens in these refrigerated cargo containers. Okay, so I kind of want to talk a little bit about communication with these stakeholders and how that's really one of my favorite parts of this job is working with these beekeepers, listening to the problems and issues that they have, and then you know, taking those and, you know, turning them into projects or for students, papers for students, and outreach and communication. So this was, I screenshot, this was a text from a beekeeper in Idaho and said, hey, what is this? And I had no idea. I mean, I guess I'm an entomologist, but I didn't know what this was and I'd never seen it before. So I asked him to send us some samples. We tracked down someone who's a much better entomologist than I am, and a guy from, he's kind of, I guess, the US expert on soldier flies. So this is a species of soldier fly that was in this colony. And he works for the California Department of Agriculture and did some imaging. And I had a new master's student, Connor Auth, who came at about that time, and kind of assigned him to this as his first writing assignment. And we published this scientific note because this was the first time that this species of soldier fly, or soldier flies in general, had ever been documented infesting honeybee colonies in the US. And so, and it was pretty interesting, you know, their groups had emailed us saying thank you for, you know, being careful about the language and making sure that people knew that this wasn't some kind of new pest, because I think this is something of economic value, the rearing of these flies for different purposes, for insect protein and that kind of stuff. This was another one that was just kind of fresh in mind. We just had the Department of Agriculture's pesticide regulatory folks, pesticide investigators out at the Othello Bee building. And so they were talking about pesticide kills. So I do get, you know, pictures and pesticide residue samples and asked to help in pesticide kills. This happened to be one from Idaho, in southern Idaho. It was like in October. They sent a bunch of these pictures and said, does this look like a pesticide kill? And To me, it didn't really, but they take the samples and all that. They sent me pictures. The ones that had kind of caught my eye were these ones. There were all these headless bees on the ground. And I saw, if you, and I don't know if I can annotate maybe. I'll try that. Let's see. I should be more used to this by now. Here, draw. Can you guys see that? Green's a terrible color. How about red? So there's a few in here that these are yellow jackets. And so when I saw this, I just thought, well, this is probably just the bees have been sitting there a long time and they've been fighting with yellow jackets, and maybe the yellow jackets chewed the heads off of all these bees. And so I told them, well, it's probably not pesticides. It's probably just normal bee death and then fighting with yellow jackets. And then almost let's see, clear all drawings. Stop annotating. Yeah, so this is just in case. So that they're headless and the thorax are all hollowed out. Something I hadn't really seen until I don't know, almost a year later, 6 months later. This photo was posted on the Washington State Department of Agriculture website as a suspected, as maybe one of the first, or might be the first honeybee colony kill caused by Asian giant hornet, but it was suspected maybe as Asian giant hornet. But you'll notice that it's the same look as those pictures from southern Idaho. So the head is missing, the thorax is all hollowed out. And so I thought, well, it's a couple of things. One is this is either not an Asian giant hornet kill over there in Western Washington that this picture is, or there's Asian giant hornet in southern Idaho. So I figured the first one is probably more true, that this is not an Asian giant hornet kill. But again, but this one doesn't have these yellow jackets in it. I had never seen this before. So until that winter, like I said, we do all this indoor storage research stuff, and this was a dead bees that are collected in a trap at the bottom of a hive. So these colonies were sealed up in a cargo container all winter, and when we took it out to look at to count the number of mites and things like that that dropped on there were all these headless bees with these hollowed out thoraxes. And so obviously there's no yellow jackets or Asian giant hornets getting in the sealed up cargo container in the middle of winter in Othello. And so then I was really confused and realized I don't know very much about anything, it seems. So in trying to figure out what this was, we found some of these in Othello on the ground. So we set up some camera traps, and we captured a whole bunch of video and images of these mice. And they come in here at night and they feed on these bees and they chewing the head off and hollowing out the thorax. So it took like a year and a half, but having these multiple different incidences of these images and these bees being dead with this similar sign of heads chewed off and the thorax hollowed out, finally kind of able to find this. And I couldn't really find anything that would help somebody figure this out on their own. And so what we did is we then published my first extension fact sheet, and this is based on distinguishing this Asian giant hornet damaging colonies. And so she kind of went through that story a little bit of assuming more people might be. So I had PowerPoint is struggling. So other people might be finding this stuff, and I figure they would be calling the Department of Agriculture thinking that their hives were killed by Asian giant hornet. And then the page I didn't put on here has sort of a dichotomous key to kind of figure out what might be causing damage to honeybee colonies. Some projects I didn't talk that much about that are ongoing or upcoming is that we're currently testing this sponsored research project with a company called HOPKINS, developing a new pollen substitute that involves like 14 different commercial operations from Washington, Idaho, California, following those colonies through almond pollination. And then VITA Bee Health, we have the Washington Commission on Pesticide Registration grant project to test the efficacy and safety of a a new product that they're trying to register for varroa treatment. It's this oxalic acid strip that's been stabilized in a glycerin solution. And then we just submitted a full proposal for an SBIR grant with Oregon State University and UC Davis and Mississippi State to look at some of these issues associated with honeybee health that tend to kind of follow them after blueberry pollination. And then we just submitted a Western SARE proposal for education and training for Spanish-speaking beekeepers in collaboration with the Bee Informed Partnership. So these are kind of additional activities, you know, so the Othello Bee Building is still relatively new and working out there quite a bit. And that's a big thanks to the commercial beekeepers that, you know, donated the money and a lot of equipment to help us build up the capacity for the number of colonies that we're trying to do out there. And then managing these gifts we have from this Altman Foundation and Northwest Farm Services credit to help get that facility up and going. And then, you know, I currently serve on the scientific advisory board for Project APSM, reviewing grants, manage a couple of service centers. So the honey that we produce is really a byproduct of the research we do. So we, you know, filter and bottle and sell the honey that we produce, and also some of the queens, and developing service centers for pollination services. So we tend to take a large number of our colonies down to California for almond pollination, and also in tree fruit pollination. We've got colonies over in blueberry pollination right now. So trying to add that as a service center as well. And then I serve on the grad admissions committee. Let's see. Yeah, so that's kind of it. I'd love to take some questions. I always try to throw pictures of my kids up here at the end, but I'd be happy to take any questions if there are some.
Thank you, Brandon. You can stop sharing, and if people would turn on their video so that we can have a conversation, super. So thank you very much, Brandon. So anyone want to start off with questions? I prefer you put your hand up, but if you're like Tim Waters and can't do that like Tim Lawrence can, you can always just go like this, Tim Waters. So Tim Lawrence.
Yeah, Brian, good presentation. A couple of things. One is that we just hired a new extension pollination person, Kelly. And how do you see you working collaboratively or with her, and what areas would you address, and what producers would you be targeting?
Yeah, that's a great question. Again, thanks for the compliment. I think, well, you know, I kind of have that advantage of her having done her postdoc in my lab, so we do work, you know, fairly well together. And so I think that's a nice advantage. I think that her background has been more with hobby beekeepers, especially, you know, in her PhD program. A lot of the stuff that she worked on with the Bee Informed Partnership and the stationary, what they call it, Sentinel Apiary program, was with hobby beekeepers. And a majority of my work, I think, is focused on commercial beekeeping. And not that she wouldn't be able to help, but my assumption would be that would be like the general split between our two expertise, I suppose, in extension. And then, like, as you know, I don't, you know, any of those are too big for any one person to handle. And so I think we would share those responsibilities as needed. You know, and because she, you know, is still just transferring out of that postdoc and into her new position, you know, she's currently a co-PI on a number of grants. One, we just got notified actually from NIFA, and so we're in the process of transferring her from a postdoc on that grant to a collaborator on in her new role.
And just to remind everybody, Kelly is not full-time extension. She has a split appointment. So Don Llewellyn.
Hey, Thanks, Brandon. I really enjoyed your presentation. Just got a quick question. I thought this deal about the imported semen was just awesome. I'm a livestock guy, and I guess you could just say your bees are another form of livestock, right? So anyway, so other than just genetic variation, to bring in the semen from elsewhere, what kind of genetics are you trying to capture with that could maybe enhance productivity or health of the bees or anything like that?
Sure, that's a great question. Yeah, and thanks. So we don't make, there's really no time really for us to make, say, any kind of selection in what we choose to collect semen from or what we're intentionally trying to import. We do target these different subspecies, and so I think in general, the goal is to get the genetics that are inherent in that subspecies as a whole that could be used to improve the population as a whole, or to select for those unique traits that are inherent in those subspecies. So like, for example, there's some new or interesting research on the advantages of this propolis envelope that bees put in their hive, the antibacterial social immune function of the propolis. And so that was largely kind of selected against in commercial beekeeping in the US, because it makes it difficult to break the frames apart and remove the frames because they glue everything together. And so then beekeepers get frustrated, they break their woodenware, all that kind of stuff. And so that was somewhat selected against. And now, they're kind of showing that there's some big advantages to that propolis. And so Apis mellifera caucasica, one of the subspecies we've collected from, is famous for, or infamous, I guess depends on how you feel about propolis collection, for their ability to collect large amounts of propolis. So that's a subspecies of great interest now to folks that that are more interested in that sort of the advantages of propolis. I don't know if that answers your question. Other than that, it's kind of just giving beekeepers a larger genetic, more tools in the toolbox for selection of their own, what they find to be most important.
Okay, I have a question. So how big is a colony? And a colony is one queen and thousands of bees?
Sure, yeah, that's a good question. So I mean, a colony could be one queen and 100 bees, I guess, in a little cage that you experimentally set up, or a fistful of 1,000 bees or something like that. So a colony is one queen and then workers in some kind of like stable state where they're producing brood and all that. But it could be as large as 80,000 bees or something like that if the colony is really strong and robust and has lots of space.
Okay, thanks. Tim.
Yeah, Brandon, I'd like it if you could also talk about an answer to Dawn's question about genetic biodiversity and the lack of it and some of the results that you've seen. But my main question is, you mentioned the new formula that you're working on for with oxalic acid and having glycerin as a stabilizer, but didn't Dr. Wu show that glycerin is detrimental to the chitin of honeybees?
Yeah, those are great questions. I'll start with a better explanation for Dawn. And so what Tim's talking about is, like I mentioned, it's been illegal to import honeybees that are semen, and they're not native to the New World. And so we've been sort of restricted to whatever genetic diversity was here when they put that ban in place, except for some, potentially illegal imports over the years, and a few by the USDA that were sanctioned by the USDA, like Russian honeybees and the Yugo bee. But yeah, so it's kind of known, and especially, if you're a cattle breeding guy and stuff, sometimes, well, not sometimes, the idea of selectively breeding is really to reduce that genetic variability. So selection and having a small number of large commercial beekeepers selecting from a small number of individuals that they graph from, reduces the genetic variability in the US population. And so I don't want to take credit for that. You know, Steve Shepard and others have, you know, measured this and monitored this and shown that the commercial breeding lines, commercial queen breeders, and the US population has declined in genetic diversity, except for you know, the Africanized bees coming into the US. And so we did not, you know, but through this importation and collaboration with commercial queen producers, Steve Shepard had a PhD student who was here while I was a student, and a postdoc, was able to measure an increase in genetic diversity in the queen producers that collaborated and participated in this importation program. So if they bought and we traded queens and virgins and this imported stock with those producers, they saw major increases in genetic diversity within their operation compared to those who have not participated in this program, saw the continued decline in genetic diversity that researchers have been seeing in the US over the decades. And then, then the other thing is, so this beta health is this oxalic glycerin thing is really fairly exciting. And I haven't seen anything published by Judy, but I do remember a discussion that the glycerin has the potential to disrupt that sort of wax barrier in the bees and maybe make them more susceptible to pesticides and other problems. So has she published anything that you know?
Not that I'm aware of, but
I do think it's a cool idea, and you know, Judy's a great scientist, so that could be possible. And that's one of the reasons why I think, you know, we have to go through these careful steps to get products registered and test, you know, efficacy and toxicity and all that stuff. The thing that I'm kind of excited about is that, I don't know if this is the format to talk about it, but beekeepers are really quick to jump on any new product, even if it's not commercially available, and maybe start testing it on their own. And so there are maybe some beekeepers who might be making this stuff up their own way in their garage and using it. And so it would be exciting to have a registered product that beekeepers could buy and purchase rather than mixing it up and making it on their own in the garage. And so we ran one trial of this fall and saw, you know, no obvious issues in toxicity. We did those Gary sort of dead bee traps and monitored you know, health and varroa levels, and the product was much more effective than the Apivar, the Amatraz registered strips, or fluvalinate, and no mortality at all. So
Okay, we're gonna move on. Ani has a question.
Hey, good afternoon, Brandon.
Hi.
I have a question about how does climate change work in your research and extension work, or does it? And do you does that come up with the communities you work with?
yeah, that's a good question. It hasn't come up that much, you know, in the, you know, say within the beekeeper stakeholders I've talked to or worked with much. But it has, I think the impacts are affecting these stakeholders. Like I mentioned, so like the high temperatures in California, the increased incidence of wildfire has a major impact on beekeepers, directly in, you know, stories of hives burning down in wildfires, them having to race out and move colonies out of the way of impending wildfire. And then but more probably impactful is like just the loss of forage. So when these like hot summers kick in early and you have, you know, 120-degree weather in Northern California, that pretty much destroys all of the forage for those bees. And so these beekeepers are having to feed supplemental food much sooner than they've ever had to before, and a lot more of it than they've had to in the past. So I think it's a major area, you know, of importance, and I think you're going to see a lot more of it. I think, directly as far as climate change, I'm involved in a group of folks from WSU and the USDA lab in Tucson. We just submitted a paper on climate change and basically how this indoor storage technique could help mitigate some of these risks associated with climate change. So it involved modeling flight days, based on temperature, and this Varroa pop model developed by Gloria DeGrande Hoffman, and showing that basically these milder winters allow for greater flight time when there's no forage for bees, and that negatively affects the ratio of nurse bees to adults, and then can cause rapid population decline in the spring, and that by keeping them indoors in this controlled temperature, it's sort of a more natural state where they're not foraging, because whenever they're flying, these bees are dying. And so anytime you have weather warm enough for bees to fly, you're losing the population, and the younger bees are aging, and they age too rapidly, basically, if you have these warm days in the winter in these northern states. So that paper has been submitted to communications biology. And so
Very interesting.
I'm going to
Thank you.
do more work on that, but that's a great question.
So other questions?
I'm just going to pipe in a little bit here, and going back to that oxalic acid comment, I see that as an important role that you can play. I remember having a conversation with a large bee breeder in California who said, I don't care what the university has to say, I care about what comes out of my fax machine. In other words, different formulas that other beekeepers are sending her. So I think having someone with credibility that can really address some of those concerns to counter some of the homemade stuff that is really prevalent in a lot of beekeeping operations is an important contribution that you can make to extension.
Well, thanks, Tim. Yeah, and I think one of the first things I'll talk about when we start presenting this data is that I have Varroa samples. It was for a different project, but in a commercial outfit, and that beekeeper had done just what you're saying. They had heard from a friend that this stuff was doing, working wonders for them. And so he mixed it up in his garage, and that's what he used for treating. This was a very, very large Washington state beekeeper. And we were we were monitoring for Varroa mites for this indoor storage stuff, and allowing him to do whatever his normal treatment regimen was. And for whatever reason, the problem with it is that there's no control over the quality of it, whether you do it right, all those things. And his Varroa mites were off the charts high, and he had to do these emergency treatments late in November, had huge winter losses because he trusted his ability to mix this up in his garage, and he had massive failure with that attempt at a product. I think having a tightly controlled, commercially produced product that's been tested and proven effective is going to be more reliable than mixing stuff up in your garage.
Okay, I'm going to change gears. Doug, I see you have your hand up, but I'm going to read one from chat. So extension faculty are required to demonstrate impacts of their programs to advance their careers. Can you describe your experience in assessing needs, developing programs, and measuring the short, intermediate, and long-term impacts of those programs?
Sure. Yeah, thanks. And Karen, you and I talked about this, and it's one of the things that I was probably most nervous with in the idea of moving to extension. And that was reminded of that again when I was talking with Tiana about this Okanagan field day that I was happy to come up and planning to come up and help with. And she asked if I wanted to have them answer questions and fill out a questionnaire about the thing. And I was like, never done that before. Why would I do that? Or what's the point? So that's not something that I have really any experience with at all. And so I would probably have to lean on you guys and have to ask questions and would have to learn along the way on how to do that. So unofficially, I mean, I can tell you that, you know, the impacts of my work, I think, you know, can be seen like in the indoor storage, for example. So when I first started doing that research, there was about, I would say, maybe 80,000 colonies that were stored indoors. And so this was a project that Steve was kind of invited to work on with Eric Olson, and I kind of picked it up and ran with it at that point, and have been, you know, doing that research and showing the and I'm not saying that I'm the one who then convinced an additional, you know, 400,000 colonies to be stored indoors, but I've been the one talking about it and promoting the and doing the research to show that it actually does improve colony health and survival. And so I guess that's one sort of way to measure impact, is that before my program, I guess there was 80,000 colonies, and now there's 400,000.
So, you know, an extension to that would be the economic impact. So we're looking for social, environmental, and economic impact.
Sure.
So I think you have opportunity with that program to do that. So Doug Walsh, sorry, I lost you on my screen.
No problem, Karen. You know, I was a few minutes late because the tree fruit entomology position kind of ran a little bit late as well. But, you know, I guess when I'm talking with the members of the general public, Brandon, I always get sort of this, they always give me this impression that pesticides are the bad guy and that's what's killing all the bees. When you get confronted with that sort of discussion, how do you address it?
Yeah, I mean, I guess I sort of address it by saying, you know, I mean, pesticides are designed to kill insects, bees are insects, and so there's no doubt pesticides can kill insects. But I don't think that they're a major problem causing these huge losses. And so oftentimes I hear that, you know, from folks that are not really in the industry, so not necessarily from growers or beekeepers, but from the general public. And so I try to just explain that things have gotten much better in the form of pesticide interactions with bees, and that the number of losses direct losses from pesticides has decreased, and that a lot of the chemistries now are somewhat softer, and that the communication between growers and beekeepers has vastly improved, I think, because of these news stories, these public stories about colony losses. And that, I mean, I think one thing is back to this idea of economics and things, and that's, I think, tends to convince people, is that when I tell people how much a grower, for example, pays to rent colonies, so like $200, let's say, in almonds, the last thing that they want to do is
Just
kill those bees. You know, those bees are a huge cost to them, and they are very, very cautious about their pesticide applications. And so, and then I just remind it, you know, that the individual people in the public go and buy pesticides to spray on their roses, and so people need to be aware, you know, of the label, and to not apply these pesticides to blooming plants in their own gardens and yards. It continues to be a good question, I think.
Other questions?
Thanks, Doug.
Okay, I'm gonna, oh, you have one? Oh, Tim, no, you just say, oh, Tiana. Tim's just going like this.
Oh, no, I just am curious, Brandon, what makes you super excited about joining the extension group versus your what you're doing now?
Well, I think that what I do now, a lot of it is extension related, even though, like I said, you know, to Chad, I think I failed that question big time, right? And that I don't have any idea how to do that stuff in a formal manner. But part of it is that my favorite part of my program right now is doing the applied research, the stakeholder driven questions, and helping beekeepers, you know, manage their operations better and find better ways to keep bees alive. And so I'm most excited that, you know, getting, I don't know what you call it, like credit for it. I don't know. So for example, a couple years ago, say an annual review, I thought I'd done pretty good. That year I'd given 36 talks to beekeeper groups and growers and things like that. But none of that, none of that really counted towards, doing a good job for the year, so it was a little bit disheartening. So I hope that, that giving stakeholder talks and extension and being out there in these sort of educational opportunities will be, I don't know, count more toward, I don't know, be looked upon more
Valued
favorably.
differently.
Yeah, valued differently. And so I enjoy that part of the job a lot, and that's not a major expectation in the current position I'm in.
So can you explain the proviso that does support you and provide some of your operation?
Sure, a little bit. Yeah, so it's been a while, but if people are familiar with the proviso, the more recent one that came through that funded the hiring of Kelly Kulhanik's position, for example, and there were some other programs that were funded out of that, like Wendy Sue Wheeler's program and stuff like that. So that was driven by the legislatures who said we need this task force. So it was the same thing, but it was, how long has it been now? 5-6 years ago. The legislature said, and Tim was on that, Tim Lawrence, I think was on, you were on that one, Tim. And some beekeepers and growers, industry groups were on this task force. They wrote up a report on why honeybees are dying. Why doesn't Washington have enough bees to even support its own pollination needs, say for apples, for example, and what the state could do to support pollinator health and support the beekeeping industry and the industries that rely on pollination. And so what came out of that mainly was the funding of this position that I'm currently funded on. And
With
it
no term.
was a bit, yeah, it was a bit odd. It wasn't so clear cut as this one, for example, because it was, that funding came through on an off budget year. So it was not considered a permanent line of funding when it was first provided to WSU, even though, they were sort of promised that basically it will be in the next, biennium budget or whatever. I don't know how that stuff works all that well. And so I was hired into this position as in a temporary manner because the funding was officially temporary when it was first came through, and then a year later it was added to the permanent budget. But that's,
So I think that's important to everybody to realize that this is not an A&R funded position or extension funded position. So I have one more question. So you have those beautiful children in your last slide. What is your interest in working with our 4-H programs, working with youth around bee and bee health?
That's a great question. I would be very interested in actually those two sons just joined 4-H a few months ago, and kind of somewhat by accident we were visiting out at Steve Shepard's place, and he had a bottle lamb, and I didn't really know, but they had my oldest son feeding it, and then by the time it was time to leave, he was crying and wanting to take the bottle lamb home, and so we ended up with a bottle lamb, and because of that, joined 4-H, and we go to the meetings and all that, and so
just so you know, that doesn't happen by accident.
yeah, and I realized that after I had to wake up at 1 a.m. in the morning and 4 a.m. in the morning and wake my 10-year-old son up to go out and feed the lamb. So I think Steve got away with one there for sure.
Right.
Steve planned that out.
Right. It's not by accident. So other questions or comments before we close?
Yeah, just one more. I don't want to dominate if somebody else has some, but you mentioned a lot of groups, beekeepers that you're working with out of state. Can just for anybody who might have a question, how that relates to Washington agriculture?
Sorry, yeah, so the question is how does all this work with out-of-state beekeepers impact Washington?
Mm-hmm.
Yeah. No, that's a good question. I kind of mentioned a little bit in that task force, part of the impetus, I think, for that one was the 5 or 6 years ago was the realization that I think apples in this state require something like 500, or maybe tree fruit in general requires something like 500,000 colonies for pollination. And I think in this state, there's less than 200,000. I don't remember what the official count is, but it's nowhere near what this state needs or requires for pollination. And so the work that I do, you know, with beekeepers in California and Idaho and North Dakota and Oregon, a lot of those beekeepers do contribute to pollination of, you know, especially tree fruit in the state, and some of them blueberries and vegetable seed in the state as well. So I think that's kind of how it works. And a lot of this stuff, the learnings from those beekeepers, you know, is in general for like the IPM is applicable to really any commercial beekeeper in the state as far as varroa control and things like that.
Okay, so we're gonna take one more question. No bites. Okay, so for those of you, thank you, Brandon. For those of you in the A&R unit, you'll receive a Qualtrics before 6 o'clock tonight. I'm gonna have a pretty short turnaround. So I think it'll be due next Wednesday, but it'll say that in the email. And for those of you not in the A&R unit that took the time today to participate, if you want to send me any comments or questions or concerns about how this might go forward, or your opinions on whether or not this guy should make the jump, just go ahead and send me an email or give me a call. So thank you very much. Thank you, Brandon. Thank you to everyone that participated again in this important process.
Yeah, thank you all very much.